Welcome, Itzik, to the Kiel Life Sciences and also to the Collaborative Research Center Origin and Function of Metaorganisms. It's a real pleasure to have you here. And you just gave a fantastic lecture. Now we have some time to talk to each other. People know you as one of the experts of the rumen, the cow microbiome. You are professor at Ben Gurion University. And developed your research over the last few decades. We know each other quite a while, and from the very first day when we met each other, I realized how much you love cows and how deep you are into them. Also, you do some experimentally challenging approaches. So, loving the cows, loving the microbiome for so many years. How did you get to there, and what induced you to the field, and when and how did it start?
Yeah. So first, Thomas, thank you for having me here. It's an amazing pleasure. So cows, it's kind of a voyage that started as by chance, I would even say. So I was trained as a geneticist for microbes, mostly studying physiology, genes, etc. And at the end of my PhD, my mentor and myself, we discussed, and it was essentially the death of microbiology. Everything was discovered. So there was no need to study any more microbes, and I was very sad about it, and I went studying stem cells. And doing that for 2 years at the Technion, one day I got a phone call from a guy in a research institute in Israel, ARO, specifically aimed at studying agricultural needs. And he was asking me about stem cells. And after this conversation, he called back. After 15 minutes, I helped him a lot. And he said, you know, there is a position here for cow nutritionist. And I said, I'm not a nutritionist, and more than that, I only understand very small things that you can hardly see. And he said, never mind, apply. And in this application process, you need to write a proposal. And it made me understand that the cow system is so complex, but yet so confined, and it holds a lot of fundamental truth that could be extrapolated throughout the world. This was the first time. So I wrote a whole proposal on how to understand microbial ecosystems. This was the beginning of metagenomics and taxonomics, what we call today. And then while writing it, I suddenly understood that for the first time in my life, I'm working in a very highly relevant ecological system that affects our globe. So I said, maybe this thing found me and I didn't find it. So you have a system of a host that highly depends on the microbiome, affects our overall environments. There are 1.5 billion cows on planet Earth, and you can actually use it and pinpoint specific host microbiome mechanisms. And this for me was a gift from, I don't know, chance, fate, and I understood that this is what I should do, and since then that's what I've been doing. Sometimes I deviate a bit, but this is a great ambition for me to actually understand the ecosystem, and during I might be beneficial for this planet and humanity in general.
Yeah, very nice. Sounds familiar. Getting to your lifelong system more or less by chance, but then stick to that. So the system found you, or you found the system. Can you summarize in a few sentences where is the system means the cow microbiome environment? Where are you now in terms of understanding that?
Yeah. So I must promote my field. So most of the anaerobic microbiology and human microbiome field was developed actually in ruminants. So the first anaerobic microbiologists, ecologists like Bob Hung, giant in the field, developed the system that people are using in the human microbiome these days. So the field from a lot of senses started to evolving before. So the ecology of this ecosystem is clear. We divide it to the specific trophic levels. We now, the past decade, using the technologies that were developed, we can actually understand that this system could be in several states that affect our globe. More methane to the atmosphere, or more milk to humanity, which is growing in population. But we still do not understand the exact mechanisms that drive the structure of this ecosystem. And I think this is where we are going next. And more than that, in this specific ecosystem, because it's very confined, we might be able to actually use this knowledge in order to modulate it. So we're on the verge of understanding the global patterns, starting to understand the more local patterns. And I hope that we're on the verge of starting to actually push them and see whether we can actually control the system.
Going a bit more specific and from a more host microbiome general point of view, looking to the cows, of course they have a very specific anatomy and system, but there are a lot of similarities also, of course, to other intestinal microbiome or gut microbial systems. And one of the intriguing aspects is that the microbiota is very dynamic in a given individual. And so when we heard a lot about the human and the newborn, which gets it during the birth process, and it changes then during life history. I think I remember a paper of you, I think it was in 2013, it's cited nearly 1,000 times now, where you follow the microbiome in a cow from the very early developmental time to the adult. So there is dynamic which seems to be general for, there is Dario Valenzano in the fish and aging, and so this seems to be a very general phenomenon. Now coming back to who drives this whole interactions, do you understand a bit about the dynamics during lifetime?
I think we do to some degree. We do understand that once we know who landed in the ecosystem, there are deterministic forces that will govern it. We know specifically for cows, because most of them, at least the domesticated setups, which are most of the cows actually live, we know what will be the order of arrival of specific species. We see that it's very, very deterministic. It's very similar for cows across the world, but we are now verifying the specific mechanisms. I think it boils down to the functions in an EU project that we're actually aiming at looking at this specific process throughout Europe and Israel as well.
So what you say, if I understand you correctly, is that there's kind of founder bacteria, and they determine then the rest of the dynamic.
Yes, and once exactly when in the verb that you use determine, I meant deterministic, meaning that one process will follow the other in a very precise and repetitive manner, specifically if we're using the same diets that are being used worldwide. Mm-hmm. And you will end up in most cases, in adult cows, you will end up at more or less at the same specific point where we can actually predict.
So people look at your work for adding to a kind of more fundamental rules in general, and so we just talked about that. But people also look to your work, of course, from a farming and agricultural point of view, and making cows better or more healthy. Is that something which is always in your mind?
Always. When we discussed at the beginning, when I understood the complexity and at the same time the simplicity of this ecosystem, I also directly understood that there is a huge ecological value in actually studying this ecosystem. So some of our findings actually show that the microbiome could be in states that are tightly correlated to the health of our globe, and specifically the amount of effect on the environment and the cows themselves, and also the amount of milk that we're getting as a humanity. So essentially, part of my quest, if you would like, would be to actually drive this process to be more environmentally friendly, more healthy to the cows, and also for humanity, yes, definitely.
We often address now the problem, and you just mentioned it, that how complex the whole issue really is, and that it is necessary that we accept that complexity, and it's not very helpful to put it down in single parts. We need a holistic understanding of the system. And then comes, so how to do that? And you talk, and you gave a brilliant talk, but you try to talk in a language that many people can follow you. In the back is completely novel technologies, in terms of wet lab, but bioinformatic. How important is that technology these days to get to the issues which we just talked about?
Yeah, trying to answer that in a politically correct way. So, from my point of view, as a scientist, always, I deem the question as way more important than any other technology. But, as I told you when we started talking, when I finished my PhD, from the point of view of microbiology, it was dead, or from the point of view of a lot of other people. Why? Because back then we could sequence one microbe at a time, or look at one proteome at a time. Today we're doing it for a whole ecosystem. So this revolutionized the field itself. So with regard to technology, it helps us a lot answering big questions, but sometimes it could be also a setback. Put all of your efforts and thinking about what the technology can provide, you're not looking above the next mountain or where you can actually go because you're confined by the limits of the technology. So that's why I was starting by saying, first you need to decide on a question from my point of view where you want to go with that. And only then the technologies actually evolve. The way to actually resolve it will come in a form of technology. So yes, technologies are important, but sometimes they can also be limiting with the way you think.
Yeah, I fully agree. I put it sometimes in different phrases, but it's also important what you say to young people who may listen to that. Question first and technology then. And it's not a good idea to have a technology and then try to invent a question where you can. Some people do that. Then they invent questions where you can use the technology. I mean, coming back to this phrase, which I learned from a mentor of mine, to learn new things, read old books. I'm sure that in your field there is probably also rich literature on the bovine, on the rumen, and the biology and the complexity of the rumen. And I assume, and I ask you, probably a lot of these papers are correct, also they cannot then come to the point where you are now. But that means they already thought and they had the question, and you now can answer it because you have novel technology.
Yeah, you're pinpointing it. For example, some of our earlier work was engaging with the invasion of the rumen microbiome early on at the beginning. This is the system that allows the cow to degrade the plant material that it eats, and it houses this complex microbiome that we're now talking about. So back then at the 50s and 60s, they had some assumption on what the microbes actually do, but they couldn't look at many microbes. They could look at the microscope and they could measure the metabolites that they are using, and the findings that we found early on at the beginning were kind of identical to their expectations, kind of looking at gravity waves of Einstein way after he was dead, and I was looking at it with awe. And these days, I actually go back to these questions because this kind of opened the direction for asking, okay, if this is the case, how deterministic is the ecosystem? Can we actually predict it? Then, for example, we can take methods, algorithms from stock exchange and actually predict forward. We actually did that when it's deterministic. So yes, I do have a lot of respect for all questions that are not answered. Mathematicians do it on a daily basis.
I know, but maybe message for us more senior people that it's so important to have time to think. And we don't have that much time. We have to fight for the time, all right? One of the key messages you told us today and also in a conference we visited recently together was your beautiful paper in Science, which shows that certain cellulose-degrading bacteria are getting lost in westernized people. Now that immediately rings a bell in many people's head, and with the title Missing Microbes. Is that something which you also worry about in your systems? And is there some ways how you think one can answer that or can fight that problem of microbes are getting lost?
Yeah. So systems, you mean the cows themselves or human beings? So cows, when they are eating what we call the feedlot feed, which is mostly corn, their microbiomes, their compositions are very different from the ones that they pasture on. But still they are exposed to certain proportions of feed that is very similar to what the cows will eat when they eat grass, when they go to herd. But still we see a lot of microbes that are different between herd animals and the ones that are living in pasture to the ones that are living in feedlots. With regard to whether I am worried about it, also for us and for the cows. So I don't know if it's bad or good. I'm worried that if we will need them again, we'll not be able to get them. I don't know if it's better or worse now. I do think, to some degree, that we can actually have methods to preserve them, exactly as we do with ecology, not only freezing them, as a lot of efforts are being done, but also keeping islands of wild domesticated or non-domesticated animals, meaning that they can grow on their old diets and keeping them in this context. What you cannot do, for example, with human beings, like you can control it to a certain degree, but you can have animals still grazing and keeping their microbes as they are without being exposed to all the other westernized or industrialized diets for animals. So this is very easy to do. I think there should be initiatives like this anyhow. So people will see a grazing cow, for example. Yeah. And once you have that, you have them preserved.
I understand your point, coming from what you're focusing on in your research. However, one of our here in Key Life Sciences, the mission is evolving health in a sustainable environment, and also in the Collaborative Research Center, we are stimulated by the hypothesis, let's put it carefully, that loss of microbial diversity and dysbiosis is correlated, and if we start to get causalities to these ever-increasing numbers of chronic inflammatory diseases, allergies and adipositas and neurodegenerative diseases and etc., etc. So could it be that the cow system this is more resilient, and we have this is in the humans, this is a worldwide problem which millions and millions and millions of people are affected, and at the moment we have this hypothesis and we try to understand what is behind, but it seems that you see that a bit different in your system. That is very interesting.
Yeah, I understand. So let's start from the humans. So the microbes that we identified in humans are specialized fiber degraders, specific polymers. And if you ask me specifically, and I'm just speculating, as you just said very carefully, I think that the fact that they cannot ferment and they are missing from a lot of human beings in the colon, they cannot ferment fiber, although we have been eating fibers for many, many generations, at least 80,000 generations. I think that it has to have a physiological effect on us. And it could be that everything that you just mentioned is due to missing microbes, specifically those. And of course, we're embarking on an initiative to actually identify these specific ones, which are extremely important. As an anecdote, most probably we got them from cows, as you probably know. This is how we started with that. With relation to cows, I would say it in a very cautious manner. I think that they might be affected by and by the diets actually that they're eating. Very hard to distinguish this from the effect of the microbes. The diet themselves with high starch are not very good when the increase of starch goes to a certain degree. But I'm not familiar with a lot of studies like ours or others that actually try to connect specific illnesses to the microbes that are missing, but more to the effects of microbes that are present. Mm-hmm. Just as an example, with relation to the starch, it's being fermented to lactic acid, which is acidic, and it affects the cow in a negative manner. So this is something that there are a lot of studies that people actually engaged in. And it could be that some of the missing microbes could actually utilize the lactate towards a different product that will reduce acidity. But I'm not familiar with a lot of studies that actually connects the missing microbes to the behavior of the cow, maybe because we are mostly human-centric with this regard.
Mm-hmm. Yeah, very interesting. Yeah. What are the challenges in the field, in your field, the black boxes, which you, of course, are now trying to address?
My lifelong quest. So I think that the field is, as I said, is in a point that there are some fundamental truths that we understand. We understand that this system is very robust. And it could be in several microbiome states, I mean composition, specific composition. We know more or less what is good, not only with broad strokes, but specific pathways that are important. And I think that we're in a point that we're trying to understand whether we can actually influence that. But influence that, people are usually using microbiome engineering. It could be that we can also influence that by understanding the forces that are governing the ecosystem. Meaning, for example, if you go to a dry land that has enough carbon, if you water it, you will have potentially trees that will grow there. But if you have land that doesn't have any carbon or any resources for plants to grow in, you will not be able to modify it. So in broad strokes, we're trying to understand how modifiable is the system and what can we do in order to change it. And in order to do that, I think one of my main quests is to actually identify what are the minimal amount of components needed in order to sustain the overall ecosystem. And once you understand the minimal amount of components, exactly like they used to do in biochemistry to fully understand an ecosystem, once you understand them, you understand what you can take out and what you can insert in order to modulate it. So I think that this is an approach that would be also good for other systems like humans or mice, but there the problem is that the system is not tightly dependent on the microbes. This is the advantage of cows.
Mm-hmm. Oh, how beautiful. Yeah, really. It's also reducing complexity to understand complexity.
Exactly. You said it in the right word, as you usually do.
Yeah, this is super. Seeing your career, and so for many you are a role model. If you think back and also think today, situation for junior researchers has changed, of course, a bit, but what's your advice to a young person who comes to you and says, Professor Mizrahi, what should I do to become a successful scientist?
So maybe I will use a word of one of my role models. It's called Yoda. It's coming from Star Wars. So he says, fear is the way of the dark side. So usually when you're creative and your ideas are not similar to others, and when others are saying similar ideas that are not similar to theirs, their usual way of reacting is rejection. So I'm saying, if you think that something is important, don't be afraid, move forward. Also, failure is not bad for you, it's good. It sculptures you to be better, learn from the failure. And our profession is about failure because most of the time we're going to the unknown. When you go to the unknown, you usually get lost. But every step that you took, which you actually made a mistake, you can actually learn from it. And most importantly, be curious. Enjoy the everyday, because these are big things that I just said at the beginning, but you need to be able to come to work, or this is a hobby for us, I would even say come to your hobby and enjoy every second of the doing. So if you are in the moment, in the doing, enjoying yourself even when you're failing, and you move forward without fear, I think you can change the world. This is how I would push, also pushing my students. So sometimes we argue, and I always end up with the sentence, okay, if this is what you believe in, you go forward and prove me wrong. So I think this should how we should educate young scientists.
Yeah, very good. As long as you enjoy what you're doing, you also are good and creative. And so that's, yeah, I fully agree. Coming to the end, but asking you to do something what one cannot do, but look in the future. And where will, if we invite you again to a keynote lecture in 5 years or in maximum 10 years, but where will the field be?
think at this point, I hope that in Hebrew we say that the prophecy was taken after the second temple. There were, prophets. So I hope that the field will be in a point that most of the functions that are extremely needed, as I said, are known to us, and we can start playing and pushing them in order to push the ecosystem to one side or the other. I will make it more pragmatic for people to understand. So I would like to be in a situation in which farmers around the world, there are 1.5 billion cows, will have the tools to actually make their cows reduce the amount of environmental impacts and supply more milk without intervening too much, but just by understanding the ecosystem. So having a red and a green light inside the ecosystem where we can actually deviate it. I really hope that we will be able to be there in 5 years. Yes.
Thank you very much. It's challenging, but maybe doable. I wish you all the very best that you get there, and thank you so much for coming to the center.
Thank you, Thomas. Thank you for inviting me.